So hi, and welcome to Module 1 of the Research Data Management for Microscopy training resource. In this lesson, we will introduce file naming conventions that you will find useful for naming your own microscopy data. And at the end of this module, you'll have had time to create your own convention in preparation for your first session on the microscope. So let's get into it. So a file naming convention provides a framework for naming our digital data. In our case, our microscopy images. And it is this framework which describes the order and the amount of information we include in the file name. By using a convention, we ensure not only that our image data is named consistently, but that the information we include in a file name will be enough such that the files are easy to find, that you and others easily understand what a file contains, and you understand how one file is related to others in your dataset. To create a convention, it's useful to think about the physical specimen to be imaged in microscopy. Many users are currently busy preparing their specimens for time in our facility. And I want you to think now how you would describe those physical specimens to a colleague, friend, boss, or even a family member. Think whether there's some defining traits or features, what we're going to call attributes, that are important to distinguish one specimen from another. Let us next consider these two laboratory mice. How do I know that the mouse on the left is different from the mouse on the right? And I hope this only took you a moment, but we can tell the mice apart because of their fur color. Scientifically, we may call this the mouse inbred or genetic strain. In this case, the black mouse is C57 black 6, and the white mouse is bulb C. Of course, not all attributes are as visually apparent. And this is especially so when you're imaging sample features at micron or submicron scale. But there are many attributes still inherent to the specimen. For example, the mice have an age, typically reported in weeks or in days. And they'll also have a sex, whether the mouse is male or female. So other attributes of a specimen may be determined by experimental design or the acquisition settings on your microscope. In a comparative study, mice may be exposed to different treatments, say a novel drug or surgery, and compared with animals in a placebo or control group. I could also image samples in my mice from different locations, say the head, lungs, tail, or legs. So attributes can be similarly identified for maybe plant and geological specimens that you acquire in the field from locations A, B, and C, which are going to have their own unique combinations of longitudinal, latitudinal, and altitudinal data. Now, these samples will be collected on a specific field trip, which may have its own unique identifier, and collection occurs on a particular date. Experimentally defined attributes in your study may include preparation of your geological or plant samples, whether they are fixed or stained with compound A, compound B, or compound C, and you may have defined parameters according to acquisition settings, such as the magnification on your microscope. Did you image the specimen with 20 times versus 40 times zoom? Now, many of our users may also perform quantitative image analysis on the microscopy data, and as such, perform image processing that affects the underlying pixel values in your dataset. You may therefore like to amend a file name to reflect changes in the image data at particular steps in your image analysis workflow. So that is just something to keep in mind. Now, the following list of attributes is also going to be available on the My Uni module page and in the file naming worksheet. I hope you find this table is useful to help describe your own physical specimens and microscopy data. So in subsequent videos, I'll show worked examples for developing file naming conventions from different scientific disciplines, including from my own research in the biological sciences. I look forward to seeing you then.